why is it that we look at technologies, and I'm thinking back to ATIA, which I know is, GNS, and the development of technology algorithms. What has been the issue in drug discovery, especially in oncology, and why is it still taking us so long, do you think, to bring new innovations to the market?
Yeah, it is funny because we were having these types of conversations 10 plus years ago, thinking that we were right on the cusp of making everything happen to really make big changes and make big discoveries in drug discovery using these types of tools. And I think ultimately it comes down to a combination of things that needed to come together and that have now come together in a truly transformative way. One is having the right data. And so we now, through all the latest and greatest innovations that have occurred in molecular profiling, as well as in the area of collecting clinical data from patients, both of those elements, at least for what we do in my current company, are critical, and I think are critical in general in AI for drug discovery, that we now have access to thanks to a myriad set of companies that work on different aspects of those data problems. So the data is part of it. Having the right AI and ML tools, and that, as you know, continues to evolve over time. Every month, there are new great tools that become available, and that everyone kind of stands on the shoulders of giants and continues to build on these AI and ML tools. More recently, it's been things such as generative AI, but it's really been this continuum over the last 20 years of machine learning and now what we call AI tools that have increasingly become available to the average user to be able to build from. And then the right compute infrastructure. Not everyone had access to hundreds of thousands of processors if they wanted to 10, 20 years ago. I remember using these physically massive machines, IBM supercomputers, that now, we have that kind of power accessible to millions of people through AWS and Google and other types of services. So it's really a combination of all of those things. And then, of course, the old school stuff is you still need the clinical and scientific expertise to be able to leverage those other three capabilities. But I think those first three things that I mentioned have now come together in a meaningful way that is materially different than where we were 10 years ago.
Yeah, it's amazing. As you said that, I was remembering back to, again, probably 20 years ago, where I sold silicon graphics workstations, right, and machines. And I remember the very first compute farms that we were building. We were building servers together. We were multi-processor, and I think 16 processors, right, not thousands of processors, was like the state of the art. Can you think back like a little bit over your experience? And I appreciate what you're saying about how these things change. Like, I think many people feel like AI, the generative AI stuff now is very exciting. We'll talk about that a little later. But in the older machine learning kind of algorithm development stuff, do you remember some like specific things that really addressed like time? Like, what was the impact of kind of like the before and after around some of the machine learning algorithms and things that happened? Because I feel like now we're at the stage where we're taking that to the next generation. But I also think we have to look at what did we really get out of the older generation of these tools?
Yeah, if I think back to, you were mentioning some of my prior experience, going back almost 20 years to working with what's now called ATIA, what used to be called GNS Healthcare. When I first met Colin Hill and Aya Khalil, who were the original co-founders of that company, going back 25 years when I met them in grad school at Cornell, they were leveraging, as I mentioned, these physically massive IBM supercomputers. They were called Blue Gene machines. And a lot of the innovation there was just being able to tailor the machine learning algorithms to take advantage of these architectures that emerged, the computational architectures that emerged. So the first set of innovation was just, how do I actually take advantage of the fact that I can use thousands of processors at once? So there was all sorts of ML algorithms about how to spread out the work. And then the fact that that was possible allowed us in that company to start looking at learning ensembles of models. So imagine you're trying to build a model of a particular type of cancer, let's say colon cancer, and you're trying to understand all the different nodes, let's say the expression of a set of genes in a network. And you're trying to get to an answer that's under-constrained. There's not enough data to learn the model that explains all of that data. So we started to take advantage of these architectures where you might have access to 1,000 processors, and you would say, let's learn 1,000 models. Each model will evolve on its own processor, and we will then use the ensemble of models together to ask a question. What happens if I knock down gene X becomes a question that we ask not one model, but 1,000 models, because we've innovated and done the software engineering to be able to take advantage of those types of architectures. So those types of things weren't possible when we didn't have access to those types of compute architectures. And then once those started to become possible, to me, that was the first wave of innovation, was just how do you take advantage of the fact that there's just fundamentally different types of computing available to us, which now seems like old hat, of course, with everything in the cloud and so on, but really wasn't back then 15, 20 years ago. And then it became a game of, okay, now that we can do that, how do we squeeze more juice by getting better and better algorithms to be able to take advantage of these architectures? And that was how that particular company evolved over time. But it started with that ability to take advantage of a large number of processors versus one processor at a time.
Yeah. So like to your point now, we've got this amazing computational capability, right? We have algorithms that have actually been tested and gotten in some ways far more sophisticated than I even expected them to get in a pretty short time period. So when we think about kind of drug discovery and the time it takes and the cost, I mean, we're still talking about billions of dollars over a period of like a decade or more to get something from, let's call it target identification, early stage lead gen, all the way through clinical trials and actually, marketing approval and then commercial launch, and then all of the like moving from an indication to another. Where do you think the big opportunities in shrinking the time scale are? If we look at kind of all the steps of drug discovery, going from that early target validation, lead identification, lead optimization, right? Initial, preclinical work, clinical work, phase 1, 2, 3 studies, et cetera. Like where's the big opportunity in your mind to kind of shorten the time to market?
Yeah, there are really multiple opportunities within that wide continuum that you just described. In the world that I'm working in right now, the ability to connect different parts of what that linear progression that you just described, I think, is really important. So in what you described, someone in a lab, perhaps in an academic environment, discovers a new piece of biology, and then they publish it. And then a bunch of people, both in pharma companies and academia and elsewhere, try to go characterize that particular, for example, drug target. It might be a protein. And so there are structural biology companies like Proteros, for example, that do that type of work and have been doing that for a long time and are masters of that. So, freeze the protein, do cryo electron microscopy to characterize it, and then, or do X-ray crystallography to characterize it. And then after that, go try to find molecules that bind that target. And there might be different kinds of physical high throughput screening approaches that take months and a lot of money and time to do. And then you try to find a hit, and then you go on down the cycle that you were describing through the various preclinical models. So make sure that it works in a petri dish, make sure that it works in animals, and then get into the clinic, and then those series of phases of clinical development that occur before the drug is approved. That process, 10 years, maybe longer. So part of the problem is that some of those pieces that I just described are disjointed. So once that target got discovered in an academic lab somewhere, it gets thrown over to that next group of people I described, and they work on it kind of out of the context of a set of patients. And so ultimately you find a drug through that long process of many years, then you go try to test it in real people. And to me, that's one of the disconnects, because at that point, you're pretty far divorced from that original exploration of the biology. And now you're saying, hey, does this actually work in a group of actual people that have this disease? And these diseases are not monoliths. So not all lung cancer is the same. Not all prostate cancer is the same. Patients are defined by the therapies that they've had before, by their genetic background, by the genetic background and the other omic background of the tumor, for example, if we're talking about cancer. And so all of these other things can get lost from that process of trying to get a drug to hit the target. And so some of the solutions that are coming through now, including some of the stuff that we're doing at Archetype, is to not lose that patient-centric focus, and in fact, start with the patient, characterize a set of patients that have a disease, and try to find drugs and screen at massive scale for drugs that will move the needle for that particular patient cohort, that will move the biology of that set of patients from illness toward wellness. And so that's a fundamentally different way to think about drug discovery. And as we're proving through some of the work that we've done that we'll talk about later, that seems to be working. And so we can short circuit a lot of the failure that's built into that process because of the disjointedness of the pieces that we described.
As you were talking, it reminds me about a project that I followed pretty closely in a group out of Eli Lilly and Company that was called the Chorus Group. I don't know if you ever ran across them, but what always struck me about them is they had this idea, it just reminds you what you were talking about, that for hard drugs to develop, for things that the risk of failure was going to be high, or which is a lot of drug discovery, especially in oncology or rare diseases, et cetera, it made sense to kind of I think in their case, they were mostly like leaving out kind of the standard parts of the process and focusing almost exclusively on this idea of proof of concept. Like if I could show that I could get from my clinical kind of academic idea to something that worked in a human being, in a patient, as quickly as possible without worrying about necessarily everything that had to happen right on a regulatory perspective, then I could go and spend all the money and time and effort to do all the, kind of heavy lifting along the way, but I would be spending that money knowing that I had a much higher likelihood of success. So I could reduce the risk. So kind of what I heard you saying in that is now that these computational methods, right, are allowing, and they were just reorganizing the structure of the project structure itself, but that we could do that kind of in a parallel way, really in silico. Am I understanding kind of where you were going, right? We could keep that concept there?
Yeah, exactly. I mean, the idea is rather than starting in kind of some esoteric biological discovery process, let's start with a set of patients that we're trying to solve a problem for. Let's characterize those patients clinicogenomically. And again, through the work of many different types of companies, that's now possible. The diagnostic companies like XCure's your company, other types of companies, were able to get access to that patient data to do that. So that wasn't possible 20 years ago. It's now very possible to characterize a set of patients, say, these patients have unmet need. We can get their clinical data, we can get their genomic data, and we can build classifiers that predict how those patients are going to fare and also describe the biological states of patients that fare well and fare poorly. And then we can say, let's start with a goal to move that set of patients from states associated with illness to states associated with wellness. So we can do that computationally. Yes, we can describe the patients, which we've been able to do for 20 years and with increasingly better data, but now we can actually act on that information using these computational approaches and say, let's find drugs that will push those buttons, move the needle for those patients specifically. And that's not to say that all the kind of canonical biology and so on gets thrown out the window. That's absolutely important, and you need that scientific and clinical expertise to validate all this stuff. But to your point, you can make discoveries really quickly. We started with a set of lung cancer patients' data, early stage lung cancer patients' data, and within a month had these types of models that I'm describing, and we're able to screen billions of drug-like small molecules, including drugs that were already clinically being tested. So one of the things you were talking about with this process with cores was, how do you get to proof of concept quickly? Well, if you're not so invested in one target, if you're more invested in validating an end-to-end approach, which is what we've done in lung cancer, for example, then you could take drugs that are already safe for people because you're not so wedded to whether that drug is your drug or someone else's drug. And you can prove that this entire approach works. You can find molecules that will move the needle for the set of patients. And you can actually test that very quickly in vitro, in vivo, in mouse models and so on. And then you can even go do the clinical trial if you have the funding to do it. We were just talking about that with one of our collaborators, Dr. Charles Powell at Mount Sinai, which is that, hey, we found drugs that look interesting. They are innovator drugs from some other company, but because they've been through the clinic, you could get right into a phase 1-2 clinical trial. And we just started this work months ago. That's really fast to be able to get that far that quickly. And so I think that's what's possible now with these types of computational approaches.